All right, so today I'm going to be talking about circadian biology. It's something that we haven't talked about yet this morning, and it's something that I'm quite passionate about, so hopefully that's going to come across. But we have talked a little bit about metabolism, and so I'm going to continue on the theme of circadian regulation of cardiac metabolism. So first of all, I just want to give a broad overview here and remind everybody that there are these marked time of day dependent oscillations in just about every aspect of physiology. Whether you're interested in cognitive function, in endocrinology, as we heard from Phil earlier, or of course cardiometabolics, which is something that I'm here to talk about today. So for example, in the early hours of the morning, there's this marked increase in both heart rate and also blood pressure. Now, classically, however, these rhythms in cardiovascular system have been attributed to factors such as our sleep-wake cycles and our feeding-fasting cycles, leading to changes in various neurohumoral factors that in turn are going to affect cardiac function. That's very much an extrinsic point of view. And so what I'm going to try and highlight today is that it turns out that there are these intrinsic timekeeping mechanisms known as circadian clocks that are found in every cell in your body. And it's these clocks that are really contributing to some of these daily rhythms that we see in cardiovascular function. What are these clocks? Well, here is an image of the mammalian circadian clock. It's supposedly a transcriptionally based mechanism. As you can see here at the heart of the mechanism, you've got two transcription factors, clock and BMAL1. And when they come together and they heterodimerize and they sit on E boxes and target genes, they will invariably induce them, including negative components of the circadian clock that are going to feed back onto that central heterodimer. And that includes the period and the cryptochromes, as well as the reverbs, the reverb alpha and beta. And they're going to repress clock BMAL1 activity. And so you've got these oscillations in which you've got an activation of clock BMAL1, then it's repression, and then it's reactivation. And it's those oscillations that take approximately 24 hours. But in addition, these circadian components right here are transcriptional modulators, and they're going to change a whole host of cellular processes along the way. So what we asked about 20 years ago was, is there an intrinsic clock within cardiomyocytes? And indeed, we found that the answer was yes. As you can see here on the left-hand side of the slide, if you put isolated adult rat cardiomyocytes in culture and you synchronize them, and then you look at the clock components over a 3-day period, you can see these beautiful 24-hour oscillations. And the timing of these oscillations is identical in vitro as it is in vivo, where here you can see BMAL1 is peaking about 12 hours before DBP, that peaks just before PER2. You see the same thing in the intact heart on the left-hand side of the slide. So at that stage we said, yes, there is an intrinsic clock within cardiomyocytes. We now wanted to know, what does it do? And so in order to address this, we made 2 mouse models in which we targeted that central clock BMAL1 heterodimer, as you can see here. In the first model, what we did was we overexpressed only in cardiomyocytes a dominant negative clock mutant protein that is able to bind to BMAL1, but it prevents transcription. In the second model, we simply knocked out BMAL1, and so now clock does not have a heterodimer partner. When you do this and then you looked at the transcriptome over the course of a 24-hour period in these hearts, you can see that those beautiful temporal patterns in control hearts are completely obliterated in both the clock mutant hearts as well as the BMAL1 knockout hearts. And in fact, using this data and a bunch of bioinformatics, we have estimated that about 10% of the cardiac transcriptome is regulated by the cardiomyocyte clock. And to my joy, many of those genes are involved in metabolic processes. I say that because my background is in metabolism, and this is what we love to study. However, knowing so much about metabolism, we know that it is a long way to go from changes in mRNA for genes encoding for metabolic proteins to go all the way to metabolic flux and ultimately to affect cardiac function. But you know what? We said, why don't we give it a go? Let's see if the clock really is regulating metabolism. And indeed it was. So first of all, we looked at triglyceride metabolism because of this guy right here. We saw that DGAT2 is a beautiful oscillation in control hearts over the course of the day that's completely lost when you get rid of the clock in the heart. And this guy is promoting triglyceride synthesis. If you look at the reciprocal pathway and you look at hormone-sensitive lipase, you see that it has the complete opposite daily oscillation. So synthesis and lipolysis happen at different times of the day. And again, those oscillations are lost when you get rid of the clock in the heart. Does this lead to changes in metabolic flux? Indeed it does. If you use radio-labeled tracers and you look at the flux for triglyceride synthesis, you see that it oscillates about 2.5-fold over the course of the day in a normal heart, and it's completely flatline in a clock mutant heart. And then when you just look at steady state levels of triglyceride in the heart, again, you see this beautiful oscillation. And you really get this accumulation at the end of the active period. And again, that is attenuated in the clock mutant hearts. Now, as was pointed out before, there is some fat around the outside of the heart. And so what we wanted to do was we wanted to make sure this was in cardiomyocytes. So we took the time and the huge expense of, I think it was 64 hearts, and we did EM studies. And we found that within cardiomyocytes, you can see that the lipid number and the area are oscillating about 2-fold over the course of the day, consistent with our previous studies. All right, so the same is true at the level of glucose metabolism. So if you look at glucose oxidation, if you look at glycolysis, or you look even at glycogen synthesis, you find that these all peak in the middle of the active period for the rodent heart, and that these oscillations are just gone when you get rid of the cardiomyocyte clock, and that's associated with a decrease in glucose uptake as shown here at the bottom of the slide. I know several of you here are interested in ketone body metabolism. This is really hot in the cardiac metabolism field. And so we noticed that this BDH1, which is a key player in ketone body oxidation, it was gone by 90% down in both of our models, the BMAL1 knockout as well as the clock mutant hearts. And when we looked at protein and even activity and then ultimately metabolic flux, they were all down. So this is an example where mRNA does go all the way to metabolic flux. Not only that, but we've been looking at cellular constituent turnover over the years. We've been looking at autophagy and mitophagy as well as protein synthesis. And that we find all of these things are circadian regulated. So you can see here autophagy has this biphasic pattern. It's got 2 peaks that are attenuated in our BMAL1 knockouts. And when we move on to mitophagy using reporter mice, we find the same biphasic pattern. You've got this increase in mitophagy during the sleep period, and then a second peak in mitophagy in the middle of the active period. Now, interestingly, when we look at mitochondrial function and we look at the individual complexes, we find that when you have high rates of mitophagy during the sleep period, it's associated then with lower levels in the activity of complexes 3 and also complex 1. However, when you switch off mitophagy at the beginning of the active period, now you can see right there you recover the activity of mitochondrial function right when you need it, when you have to have that increase in ATP production, when you wake up and you have that increased energetic demand. And when you get rid of mitophagy in the heart by using the Parkin knockouts, you completely lose these oscillations in mitochondrial complex activities. All right, so just to summarize what I've shown you so far, I've shown you that at the beginning of the active period is a time of increased oxidative metabolism. And we think that is there to meet that energetic demand for when you first wake up and you have to have that increased contractility. At the end of the active period, that is when you take those excess nutrients that you've been consuming during the day, or you might have had a big dinner, and you then store that in the form of triglyceride. As well as glycogen. So you've got that energy that you need during the sleep period when you're fasting. Because when that sleep period happens, that's the time of cellular constituent turnover. That's when you have to do your preventative maintenance, and you've got to get rid of damaged cellular constituents and make new ones so that when you next wake up, then everything is working completely normally. All right, so that's a little bit of the history of what we've been doing. And so what I wanted to talk to you today for the rest of the time is the potential mechanistic links between the cardiomyocyte clock and cardiac metabolism. Basically, how does it do it? I mean, there are so many transcription factors in the clock, and I told you it's regulating 10% of the transcriptome. can we start to narrow down some of the key ways in which the clock does this? And that's what we've been trying to do. We've really been focused on these three genes that I'm showing you right here. This PIK3R1, NAMPT, and DGAT2, because these regulate insulin signaling, NAD metabolism, as Maria mentioned earlier on today for NAMPT, as well as DGAT2. I already talked about that for triglyceride metabolism. And when you get rid of the clock in the heart, these things just go away. They become arrhythmic. And so what I'm going to do is I'm going to focus mainly on NAMPT and DGAT2 because for PIK3R1, after we did some reporter assays using some promoter bashing basically, we clearly showed that clock and BMAL1 bind to the promoter and activate this gene. But after that, we really haven't done much else, and neither has anybody else. So what I'm going to do is I'm going to focus on NAMPT. Now, this is a very busy slide because what I'm trying to do is I'm trying to show a lot of data from not just my lab, but also the laboratories of Mitch Lazar, as well as Mukesh Jain, and also Lili Zhang. So between the four of us, we've been all focused on NAMPT and trying to understand how the clock is regulating this particular very important gene, leading to oscillations in NAD levels, as you can see here, over the 24-hour period, and NAD levels are elevated during the active period. So here's the model based on all of this data, which includes reporter assays, it includes ChIP-seq, it includes multiple knockout animals. Basically, I'm summarizing all that data on the right-hand side of the slide. What you can see here is that during the sleep period, clock and BMAL1 as well as KLF15 become activated. And they, in turn, will activate the NAMPT gene, such that then you have a peak level of expression of NAMPT right at that transition of the sleep to awake. So right when you wake up, NAMPT levels are highest in the heart. And that is correlated then with an increase in NAD levels in the heart, and that is going to be used for oxidative metabolism at that time. And then as you go into the active period, you have to have a break on this gene. And so this is performed by E4BP4, this transcription factor here, which is now going to switch off NAMPT as you're awake so that you can have this 24-hour rhythm. All right, so that's NAMPT. That's what we know about NAMPT. Here, I'm now going to start getting into not just transcriptional, but also post-transcriptional regulation of this guy here, DGAT2. So first of all, when we were doing the studies with NAMPT, we also noticed that DGAT2 is a direct target gene of E4BP4, which is regulated itself by reverb alpha, and so we're pretty happy with this axis right here. But more recently, we started to look at microRNAs, and we decided to ask the question, does the clock in the heart regulate any microRNAs at all? And in fact, the answer is yes. We identified a whole host of microRNAs that are regulated by the clock, and we think many of these are regulated directly by reverb alpha. One of them caught our attention, though, this guy right here, LET7C, because LET7C, as you can see here, is regulating a whole host of circadian clock components. And so we think LET7C might be another feedback loop in the clock, another way in which the clock self-regulates itself. But in addition, it's known that LET7C regulates a number of metabolic genes, or mRNAs, including DGAT2. So we went on and we looked at LET7C, and you can see here it's a moderate oscillation in control hearts, and in our knockout hearts, you can see that it's just chronically elevated. When you do the same thing in a reverb alpha beta double knockout heart, you also see LET7C is elevated. And in those double knockout hearts, you find that DGAT2 is also lower as well. And we think that LET7C is contributing towards this repression. All right, so that is in the title of my talk, the transcriptional and the post-transcriptional regulation. What I wanted to move on more towards is the post-translational regulation, these types of elements for the circadian clock. Because it turns out that circadian clock components are not just transcription factors, but they don't just bind to DNA, but they bind to other proteins in the cell. And we know this largely from work in the liver. So here you can see that in addition to the mRNA for BMAL1 oscillating, you get a modest, about twofold oscillation in BMAL1 protein. And in the liver, this is known to bind directly to various factors involved in protein synthesis, so transcriptional elongation and initiation factors, which is kind of interesting because we do see that protein synthesis does oscillate over the course of the day in the heart. However, that was just about a twofold oscillation. I didn't get too excited about that, but instead this guy here, reverb alpha, at the protein level, you can see that over the course of the day, this is changing tenfold. That's a huge oscillation in a protein, and that, again, work in the liver has shown that this can directly bind to and affect the activity of OGT, or O-gluconate transferase, which in turn is going to affect post-translational modifications of proteins through O-gluconacylation. And finally, we've got this guy right here, PER2, which again oscillates quite nicely over the course of the day. It's about a 6-fold oscillation in the heart. And again, work in the liver has shown that this can directly bind to and affect the activity of mTORC1. And so it's these two that I wanted to focus in on for the rest of my talk. So going to this O-gluconacylation, many years ago we had shown that in the heart there's this nice time of day dependent oscillation in total protein O-gluconacylation. And in fact, many circadian clock components themselves are O-gluconac modified, and we believe that this is another feedback loop, a little bit like that microRNA that I mentioned earlier on. So when we saw some of these more recent studies come out, we decided to ask the question, how important is this oscillation of gluconacylation for changes in metabolism over the course of the day? So we teamed up with Adam Windy at UAB, who has a mouse model in which you can increase protein O-gluconacylation only in cardiomyocytes in an inducible manner. This is a dominant negative OGT cardiomyocyte specific model, and after 2 weeks of an increase by only 50% of O-gluconacylation in the heart, we find that we completely lose all clock output in these hearts. It's completely gone. So you can see here that these beautiful time of day dependent oscillations that you usually see in a control heart, it's just completely wiped out in these dominant negative OGA hearts. And if we look at my 3 favorite metabolic genes, DGAT2, NAMT, and PIK3R1, you can see that these oscillations in control hearts, they're just flatlined. It's almost like the clock has been completely obliterated in these hearts. All right, so moving on to PER2. So as I mentioned, that there were some studies about maybe 7 years ago showing that PER2 can directly bind to mTORC1. Well, more recently in the cancer field, this group was very interested in the idea that PER2 might be binding to other proteins in cells. And so utilizing a proximity assay, biotinylation proximity assay, they found a whole host of proteins that PER2 is putatively binding to. When I went and mined that data, I found that there were at least 15 metabolic proteins that PER2 is potentially binding to, as you can see in this table right here. So what we did was we took 4 of these proteins, and we first of all confirmed that in our BMAL1 knockouts, there's no effect whatsoever on the mRNA. And we looked at by Western blot at those 4 targets, and we found that all 4 were about 2-fold higher in our BMAL1 knockout hearts. So this is completely independent of transcription. So we were really excited about this, and so we said, okay, let's go and invest in making a PER2 knockout. It hadn't been done before, so we've made a cardiomyocyte-specific PER2 knockout. And when we look at this first target right here, fatty acid synthase, which is increased when you get rid of a positive clock component, that's BMAL1. If now you get rid of a negative clock component, PER2, you see that fatty acid synthase levels go down, the complete opposite. Not only that, but when we perform metabolomic studies, we find that it's very clear that in our knockout hearts, the metabolome is very different compared to control hearts. So we think PER2 is going to be a very important regulator of cardiac metabolism. Okay, so just to summarize, here's what I've shown you today, that there is within a cardiomyocyte this so-called circadian clock mechanism that has been considered to be primarily transcriptionally based, and we found over the years that it regulates a whole host of mRNA species that in turn is influencing cardiac metabolism. But more recently, we're finding that the circadian clock is regulating a whole host of microRNAs as well. And not only that, but these clock components can directly bind to other proteins in the cell, and together, through all of these different mechanisms, the clock is coordinating temporal control of cardiac metabolism. So with that, I'd just like to thank all my collaborators for all the help over the years, and I'd like to thank you for your attention. Thank you very much.